Hello, everybody, and welcome to our October Quest lecture. For those of you who don't know me, my name is Jeanette Bruce. I am the program coordinator at Whistler Public Library, and I'm delighted to be hosting this evening's talk on behalf of our friends at the Squamish Public Library and the Pemberton and District Public Library. It has been a really great pleasure to be able to partner with all the Sea to Sky libraries for this series since the summer, and of course to continue our partnership with Quest University and all the wonderful people there. Before we get going, I want to express my gratitude that we're able to gather tonight virtually on the shared traditional unceded territories of the Squamish Nation and the Lilwat Nation. We are very lucky to live, work, play, and learn on these territories. For those of you who are joining us on Facebook, welcome. We will be taking questions from Facebook after all of the presentations tonight. And I will be monitoring that feed. So please do feel free to leave your questions in the comment section on Facebook. That's if you're watching us live, of course. If you're watching after the fact, you may be out of luck, but we do hope that you still enjoy the presentations tonight if you're watching us in the future. So I am not going to do too much more talking because we have not one, but four guests this evening. It's a novelty for the Quest Lecture Series, and I'm excited to see how it goes. But I'm going to pass you over to someone who may be a familiar face. Dr. Marina Tourlakis was one of our presenters in the summer Quest Lecture Series. So you may, and actually she's presented for that as well. So you may recognize her, but I am going to pass it over to her right now because in fact, the other presenters tonight are her students. So Marina, take it away.
Wonderful, thank you. And thank you very much for inviting us to give this talk. It's really quite fun that we get to not for me to talk. It's the best situation where I just have to be here and cheerlead, and my students will do the work. It's basically what every professor hopes and dreams for. So welcome. My name is Marina Tourlakis, and before we begin, I'll just echo Jeanette and ask everybody to just take a moment to reflect on where they are. We, and by we I mean mostly me, I'm on the land of the traditional and unceded territory of the Squamish peoples. Quest University, where I work and where my students are also attending, sits on the Squamish traditional territory, and I am grateful to have the opportunity to work, learn, and live in this blessed territory. And again, I encourage everybody to take a moment and think about where they live and what that land is and who that land is originally belonging to. So today we have three students plus myself. My name is here in case anybody is not catching the pronunciation, and I also have it here in case you want to look me up and continue this conversation outside of this talk. And essentially what we're going to do is I'm going to give a very brief introduction about why we're here and who our three panelists are. They will then give their talks, which are about 10 minutes long. And then at the end of their talks, we will entertain any questions and have a discussion about those talks. So by way of introduction, basically, there's three motives that I wanted to relay in terms of why we're here and what I had in mind in coming up with this idea for this talk. So one was to provide a space for a community conversation about local and regional concerns, which have to do with things like climate change and food security, but to think about that from the perspective of biotech and how plant biotech might be leveraged to address some of those concerns. This was in part to mirror a panel that perhaps some of you in the audience may have attended, which was on Monday, which was more geared towards, well, it did have some biotech feature, but not so much. And today we'll be more or less talking about that. Another motive was to highlight how studying and working with plants which is a deeply underserved and underrepresented branch of biology, is really important, especially today in terms of finding solutions for grand challenges such as climate change and food security. And finally, and perhaps most importantly, I wanted to offer an opportunity for students to share their work with the broader community and to gain experience with science communication and with answering questions from the public. So hopefully we get some questions from the public so that they can indeed practice that. I think these are really important skill sets for students to learn, particularly today in our society where we have a bit of a crisis of evidence and well, let's say alternative facts and things like that are floating around. And I think now more than ever, building science communication skills and building discourse skills around controversial topics is really, really quite important. So joining us today are 3 plant enthusiasts who just completed a course with me. The course is called Plant Development and Biotechnology. And in this course, we learn about plant development mechanisms, how we study plants, how plants interact and cope with a sometimes hostile environment, particularly given that they cannot move away from that hostile environment, and how cutting edge technologies in the academic and industrial lab can be employed to understand how plants work and also to confer desired traits in plants in a specific and efficient manner through things like gene editing. The major project in this course asks students to choose a specific area of focus for research. They are tasked with developing with generating a literature review on a topic that is relevant to the theme of this panel, how plant sciences can be leveraged to meet today's grand challenges. And then they are asked to translate a portion of that written work, which is geared towards an expert audience, into a short presentation that's geared towards an educated non-expert audience, which would be something like a public setting like this. So first, we will hear from Umair. Umair Milky joins us from Richmond, BC. His academic focus at Quest centers on the question, what stories do genes tell? And he is interested in pursuing a career in genetic counseling or research after completing his undergraduate and graduate education. Secondly, we'll hear from Tristan Jaffray. Tristan joins us from Colorado Springs. His academic focus at Quest centers on the question, how do we engineer urban ecologies? And he is interested in merging disciplines such as civil engineering and wildlife management in order to actually understand the human-created world. And our final speaker today will be Kenya Sengor. Kenya joins us from Victoria, BC. And Kenya's academic focus at Quest centers on the question, how should we feed ourselves? He's very interested in physiology and exercise physiology and molecular biology and hopes to attend a master's program to become a physiotherapist. So we will hear from each of these speakers for about 10 minutes each. If you have any questions, we ask that you save those to the end, but feel free to drop them down in the chat or through Facebook. And once we have completed them all, we will take a look and see and hopefully have a rich discussion. 2 of the talks are pre-recorded, so hopefully you'll forgive me while I awkwardly stop sharing my screen and then re-share my screen to go to a different talk. And then the last one will be a live presentation. Okay, so here we go. The first talk again is from Umair, and bear with me. Okay, are we seeing it? Sweet. Oh, just a second. It looks like it wasn't at the beginning. Okay, here we go.
In 1986, Grimsley and his colleagues
Hi, folks. I'm just going to jump in for a second because we're having a little bit of a technical difficulty. Marina, we can't hear while you are muted. So can you go back to the beginning and start again?
In 1986, Grimke and his colleagues described a method to study plant-virus interactions. Today, we are using this method to produce a potential COVID-19 vaccine. Wait for it. In plants. How frigging cool is that? But one might wonder, growing pharmaceuticals in plants, what have you been smoking all day? It's simply not possible. But let me tell you, it's actually not only possible, it's quite simple using agroinfiltration. What? Agro what? Oh, well, let me tell you all about agroinfiltration then. But first, I would like you to imagine a chemical reactor for the production of pharmaceutical protein. Big, heavy, with all its wires and multiple compartments that need to be kept clean. Now imagine all the money that must have gone into building this thing, and all the money that goes into maintaining it. Now, imagine a small plant, such as this one right here on the screen. Imagine if this plant could also produce pharmaceutical protein. Except now, the cost of the production of pharmaceutical proteins would be reduced by roughly 12 times, because the cost of maintenance of a plant this small is nowhere close to the cost of maintenance of a huge chemical reactor. Advancements in gene technologies allows us to insert genes coding for the pharmaceutical proteins we are interested into the plant cells. This is called molecular farming. But how do we insert our gene of interest into the plant? Agrobacterium tumefaciens, pictured on the left right here, is a natural plant pathogen which induces tumor in plants by inserting a part of its tumor-inducing plasmid, pictured right here on the right. The region that it inserts into the plant cell is the tDNA region. Because of its ability to insert part of its own genome into the plant, it is often referred to as a natural genetic engineer. Scientists have exploited this natural method and inserted their gene of interest into the TI plasmid in place of the tDNA. Then they just introduce the bacteria into the plant cells, and well, the bacteria do what it does best. But how do researchers get the agrobacterium into the plant cell? Why? By using agroinfiltration, of course. Owing to its simplicity and efficiency, agroinfiltration is a popular tool in the field of biotechnology and plant biology. It was initially developed as a tool to study plant-virus interaction in 1986 by Grimke and colleagues, and it has since then been adapted to be used in molecular farming to yield large amounts of pharmaceutical proteins in plants. But how does one go about infiltrating a plant cell with agrobacterium? Well, there are two ways you can do that. However, let me first introduce you to a plant species that is used in agroinfiltration rather often. Panel A here shows Nicotiana benthamiana. I'm pretty sure I butchered the name. Well, these plants are relatives of tobacco, and they are often used in agroinfiltration and they can yield high amounts of biomass rapidly. Figure B here depicts syringe infiltration, and it does exactly what the name suggests it might do. In syringe infiltration, we take the infiltration medium containing the agrobacterium and create a small niche on the back of the side of on the back side of the leaf and inject the infiltration medium into the leaf through the niche. As the infiltration medium enters the leaf, the color of the leaf darkens, as shown right here on the far right. Syringe infiltration is simple and allows us to introduce several genes of interest in the same plant, or you can introduce several genes of interest on the same leaf but in different areas. However, not all plants can be infiltrated by syringe infiltration, and we have to resort to vacuum infiltration, which is depicted right here in panel C. It involves submerging the plant leaves into an infiltration medium containing agrobacterium strains. The submerged leaves are then subjected to a negative atmospheric pressure in a vacuum chamber. The vacuum draws air out of the interstitial spaces in the submerged plant leaves. When the vacuum is released, the air spaces are occupied by the agrobacterium strains. Vacuum infiltration allows us to rapidly infiltrate a plant, but it does not provide us with the flexibility that syringe infiltration does. However, vacuum infiltration is extremely popular in molecular farming due to its enormous scalability potential. But how do we insert our gene of interest into the bacterium to begin? Good question. I call this playing with viruses. Basically, to allow for quicker expression with higher yield, a vector usually based on plant virus is designed to carry the gene of interest. These vectors are then cloned in E. coli bacteria as depicted in the slide right here, which divides rapidly and therefore allows for a rapid cloning process. The cloned vectors are extracted and electroplated into the agrobacterium infections. Agrobacterium containing the vectors are then introduced into the infiltration culture, which is a mixture of various chemicals functioning to ensure efficient expression of our genes. Well, what happens next, you say? Before we can introduce the infiltration culture into the host plant cells, we have to grow the plants in a set of uniform conditions to ensure maximum protein yield, as shown in panel C over here. These conditions vary between different plant species. Usually, when the plant reaches an age of 6 weeks, agroinfiltration is performed as shown here in panel D. Who would have thought that making pharmaceutical protein can be so much good? Agroinfiltration has been used to harvest many recombinant proteins in the plant over the years. Even though agroinfiltration allows us to produce high amounts of proteins, these proteins are not always effective. One of the only plant-produced proteins to have been approved for use by the FDA is a therapeutic enzyme for Gaucher's disease. Another protein, ZMAP, which was expressed in tobacco, was the first treatment for the Ebola hemorrhagic fever during the Ebola epidemic in Africa in 2014. Later on, however, ZMAP was found to be inefficient compared to other treatments that followed. Remember how, at the beginning of the presentation, I told you how we are using plants to produce a potential COVID-19 vaccine? It's true. A Canadian company, Medicago, is developing COVID-19 vaccine using vacuum infiltration in Nicotiana benthamiana plants. As of August 2020, this vaccine has entered the first phase of clinical trial. Medicago has also developed an influenza virus vaccine, which is currently in the third phase of its clinical trial. So, even though it might feel like fiction, agroinfiltration and development of other biotechnological processes have really paved the road for rapidly producing pharmaceutical protein in a plant in a cost-effective way, as maintaining plants are relatively cheap. This is super important, especially in cases of pandemics and epidemics, as it allows developing countries with an option to mass-produce vaccines at a very low cost. In an animal privileging world, plants might finally be starting to get their dues, even if it is for human benefit. Thank you. Just wanted to leave you with this because don't forget, plants are super dope. Wanted to say it at some point and thought this would be the best place to do so. Thank you again.
Awesome. Every time I see that last bit, I chuckle. Wonderful. Okay. So a little bit about how we can use plants to grow drugs. All right. Wonderful. Next up, we have Tristan. So let's see. Sorry about the audio glitch. If I was really clever, I could probably figure out how to make sure that you can't hear me, but you can hear that. But I'm not going to improvise right now. We're just gonna run with this. Okay. All right. Here we go. Second talk. Desert fruit GMOs.
Hello, my name is Tristan Jeffrey, and today I'll be presenting on desert fruit genetically modified organisms. Climate change drastically alters the conditions in which crops are raised, but research fails to illuminate the possibility of genetically modifying foods in order to resist the negatives of heat stress. Historically, temperature has been critical to the development and growth of plants, especially crops. Every historical story has something about, dry summer, droughts, early winters, the cold. We just know that temperature is critical to plant development. We also know for a fact that global temperatures are rising. So this poses a challenge for agriculture in getting plants to respond better to rising global temperatures and to a warmer climate. And plenty of research has gone into showing how we can make certain crops genetically resistant to this or other agricultural practices. We've seen a lot of research go into genetically modifying things such as wheat or corn to grow in warmer climates and to be more heat stress tolerant. And we've seen quite a bit of research in other agricultural practices such as shading or watering practices that can help fruits deal with rising temperature. But there's a gap in our knowledge for fruits that are being genetically modified to become heat resistant. As far as I can tell, there's almost nothing. So in this study, I'm going to first highlight what heat stress is, how it works in fruit, what that might look like. Then I'll dive into some of the mechanisms behind the heat stress response system. And lastly, I'll be discussing how various studies that have been done before show that this gap in our knowledge can totally be filled, and it's possible to see, how we can start to genetically modify fruits to be more heat resistant. So when we talk about heat stress, we're talking actually about a lot of things that all work on the same mechanisms and pathways in a plant. Heat stress can be caused by changes in temperature, of course, but heat stress can also be caused by things like drought, soil salinity, too much light, and can even be caused by various viruses and bacteria. It's hugely complex, and because of this, plants both have a number of ways of dealing with heat stress, such as shedding leaves or extra extremities, changing their chemical composition, or upregulating proteins that can help with heat stress. And it also means that when a plant can't handle heat stress anymore, it can have a wide variety of consequences as well. So it's a large topic. And we can see that here in this table of Sharma and Manjeet's review, which this is a subset of 26 different fruits that they examined. And here we have the names of the fruit on the right, or sorry, left, and we have the effects on the right. And you can see that these vary hugely. In strawberries, too much heat just causes albinism, but in a prickly pear, overheating results in death. So because there are so many phenotypic results that can happen from heat stress, it can be really hard to create a genetic modification that works with a plant to prevent it. We don't really know what we're looking for when we're trying to prevent heat stress. It's hugely complicated. Fortunately, we can start to better understand heat stress and combat it by understanding the mechanisms behind it. So here on the right, we have this huge, messy flowchart, which just shows part of the mechanism responsible for addressing heat stress in a plant. At the top, we have the heat stress, and at the bottom, heat stress can either have long-term or short-term responses to heat stress. And here in the middle, we have all the different proteins and ions and everything responsible for making sure these changes happen in a plant. And what Ohama et al. Did, the creators of this figure, looked at is the HSF-A1S protein, which which you can see right in the middle there. And by examining this protein, these researchers were able to find that even though this huge system is entirely messy and complex, the HSF-A1S protein is basically the master control, the control panel for all of this. It's central to both short and long-term heat stress response. And pretty much any time there's too much heat for the plant to deal with, we see more HSF-A1S. And this has been confirmed by other researchers like Kyan et al., who when they upregulated this HSF-A1S, they saw everything else change. So even though this is hugely complex, and this figure is only a small part of the heat stress response, and some of it is still up for questioning. We don't actually know whether it's calcium ions or ROS that cause the activation and stabilization of some of these proteins. We still can look at this mechanism and say, okay, here's some changes we can make to a plant to help it deal with and regulate heat stress response. Interestingly enough, this heat stress response system evolved early in organism history and is shared not only between almost all plants, but we've even seen some examples of this response mechanism in mammals. It's a hugely critical structure, and because of that, changes to it just don't happen that much. So we know that when something here works in one plant, a change will probably work the same way in another plant because it's pretty much shared across a huge number of organisms. Now it's mostly just the genes that the system expresses that change from plant to plant. So a couple of experiments have explored this direction. The first one looking at tomatoes didn't genetically examine them, but it did examine this HSF-A1S protein in response to heat stress. And what they found is that tomatoes that upregulated this and dealt with heat stress, their fruit became much more viscous, and they lost much less water because so much of their juices had been stored in much more viscous membranes. And so what this tomato is doing is it's increasing the ion count. It's making its cells more salty, and so that it can retain a lot of that water. And that process is caused by this HSF-A1S protein. And so they can see, like, listen, in heat stress, this is how tomatoes deal with it. This is what it causes. We can also look at other changes to the HSF-A1 system and the heat stress response system. In another study, he et al. Looked at wheat and Arabidopsis, and they looked at a specific protein, TAWRKY1 and TAWRKY33 protein, which is just part of that huge cascade you saw earlier. And what they saw is that when they introduced genetically this protein to be upregulated in wheat and Arabidopsis, they saw a much greater increase in heat tolerance in these plant species. They saw that they lost a lot less water, and the produce that they produced suffered a lot less from heat stress responses. Finally, our third and most critical study for this was looking at vanillic acid in blueberries. And while this wasn't genetically implemented, what Ann et al., these researchers did, is they treated roots with mixtures of vanillic acid, another important transport protein in this whole big mess of heat stress response. And they saw that blueberries treated with this vanillic acid were basically 4 times as productive under heat stress as other blueberries, and used much less water, dosing about 40 milliliters per gram of this vanillic extract in the root structure was basically able to massively increase the blueberry's heat tolerance. And of course, blueberries are very difficult to grow in warm climates. So we can look at these 3 studies, 2 of them with fruit and 1 of them with genetic modification, and say, like, okay, we see that this one big system is shared between all of these plants. And we can see how changing specific aspects of it very carefully, we can get the desired heat stress response factor. But genetic modification hasn't been tested yet. There was no genetic modification in the tomato study There's no genetic modification in the blueberry study, but we saw genetic modification work in the Arabidopsis and wheat. That brings me to my conclusion. We know that modifying heat stress proteins in fruit is possible. We know that modifying fruits to be resistant is possible. However, the system is very complex, as we've seen, and we still need more studies before we can get to that step of it being commercially viable. We're so close. We just need 1 or 2 good studies on, showing that fruits can be modified to be heat stress resistant, doing the same things they did in he et al. Study with the tauruki, with changing or upregulating HSF-A1 protein, and we'll be there. So we found that heat stress can have multiple effects. Fruits can respond in wild ways, but the central mechanisms behind this are shared between all plants. And by changing that system as a control panel, we can create heat stress tolerant plants. This isn't imagination. We're just a few experiments away from commercially viable heat resistant fruits.
Great. Wonderful. Okay. So that was the talk from Tristan, learning a little bit about how plants can handle a hot climate and a heating up, that's not words, warming up climate. And an interesting teaser that actually will nicely lead into our next presentation, thinking about how we might possibly modify plants to help tolerate such changes in climate. So our final presenter today is Kenya. Kenya, I believe you're going to share your own screen. Is that right?
Yeah, I will.
Okay, wonderful. Okay, so then I'll let you take over.
Okay. Oh, excuse me. I have to authorize the use of my camera real quick.
Wonderful. It's possible you might need to restart Zoom. We'll see.
Excuse me.
We will wait with bated breath.
Okay. Excuse me. I'll have to leave and I'll rejoin within 30 seconds.
I will take this opportunity to remind our viewers that we will be taking questions both on Zoom and on Facebook. We are going to wait until the final presentation is done. So once Kenya comes back, we're going to wait until all 3 are done, and then we will tackle some questions. As Marina knows, I always have my own list, the length of my arm, but we are definitely looking for questions from the public. So whether you are on Zoom, you can use the Q&A function or the chat function. Either way, I'll have a look at both. And if you are on Facebook, you can comment on the video, and I'll be monitoring over there as well. So, and I think that my list, just for the audience, my list, I've got some questions for individuals and then some questions for everyone, including Marina. So, not anything goes really. You're welcome to ask nitpicky questions about the presentations, general queries about plants. Oh, hang on. I think Kenya might be a participant. Promote to panelist. Here he comes. Welcome back, Kenya.
There we are. Sorry about
that.
Sorry, you were an attendee, and I had to make you a panelist.
Gotcha. Thank you so much.
No problem.
Okay, let's make this work now. Can you see my screen?
We sure can.
Okay, awesome. Well, I'm here today to talk to you about some of the applications of plant gene editing in addressing climate change. So let's get into it. Well, we find ourselves in a bit of a predicament. Climate change is set to drastically reduce the amount of land that's available to us for farming. And at the same time, our population is growing such that we may have as many as 10 billion people by the end of this century. And so our most valuable crops, crops that we use for not just food, but materials and for their pharmaceutical properties, our yield of those crops will have to increase quite significantly and quite quickly. And while conventional means like phenotypic selection of plants for desired traits might help us to some extent in order to produce desirable traits like stress resistance, it may not be able to do so within the time frame that we need it to. That being said, technologies like gene editing may be up to the task, and they're quite powerful technologies, and they allow us to do some incredible things that conventional means would not allow us. That being said, there are some barriers to their use as of today. So in many cases, regulation in many countries is deeply out of touch with the science, such that it's not really well informed by where the scientific consensus is on the technology. And this is accompanied by a pretty deep distrust in the general public. And this might be attributable to the fact that these technologies are relatively new, and they are very, very complex. And so this has produced quite a difference in the standards surrounding genetically modified commodities and organisms across countries, such that international trade of genetic modification technologies and genetically modified commodities has been hampered. And because trade isn't really on the side of this technology as of now, this has led to a pretty significant constriction in the funding and the research being directed towards genetic modification technologies. And so these technologies are really being held back from their highest potential. But hopefully, by illustrating some of the basic mechanisms and applications that these technologies can have, I can kind of quell some of that distrust that some of us might be harboring towards this technology. And maybe we can begin to think a little bit more about some of the more profound applications of these technologies. And so while Umair spoke about agroinfiltration or agrobacterium-mediated gene transfer, my presentation originally contained a section on this. I've chosen to skip over that because he already covered it in such excellent detail. And so I'm here to talk to you about CRISPR-Cas9 instead, which is another really promising technology. I'll just get into it. Essentially, CRISPR-Cas9 is based on an existing immunological system found in nature, shown by bacteria when they encounter viral DNA. When viral DNA enters the bacteria creates a strand of RNA, which forms a complex with an endonuclease called Cas9. And essentially, that RNA attaches to a matching section of the viral DNA, and Cas9, being an endonuclease, initiates a double-strand break in that DNA. The cell then tries to repair that DNA by initiating a process called non-homologous end joining, which if you look at the diagram is on the left-hand side. And this tends to produce deletions and insertions, which generally cause the inactivation of that gene. That being said, scientists have realized that through carefully constructed RNAs, or carefully constructed RNA and Cas9 complexes, they can steer pieces of RNA which are accompanied by pieces of donor DNA to any section of a genome in order to make targeted changes. And this exploits a second repair pathway called homology-directed repair. In the presence of a homologous piece of donor DNA, homologous meaning that it matches the sections that have been cut, in the presence of a homologous piece of donor DNA, that donor DNA will then be affixed in the area which was split open by Cas9. And so this system, when we carefully construct guide RNAs, can be used to direct very specific and accurate alterations with little to no unpredictable mutations to any cell in any living organism. And this has some really profound implications. This is a relatively new technology, and so we don't know everything about what it can do, but we've begun to imagine some really profound things. And so here's an example of how scientists might put this system into action. Please don't be intimidated by this very busy graph. Basically, or this very busy schematic. Basically, what this is showing is in one study, scientists were looking at a cotton plant, and they identified two genes, which you can see at the top of the schematic. They identified these two genes as having a number of downstream effects on a variety of other genes and proteins. And that's what you see in the rung below. And below that, you can see what sort of physiological effect comes from those proteins and those enzymes and those genes which are affected by the two genes in question. So essentially, they targeted these two genes in order to promote their overexpression such that they would synergistically cause all these downstream effects and bring about greater abiotic stress tolerance. And so the results of an experiment like this particular diagram is from a study on rice, but it shows kind of what sort of results scientists would see from an experiment like this. Essentially, on the left-hand side of this diagram, what you're seeing is, if you look at the bottom, there's a part that says NT, that means non-transgenic. It's referring to, in that column, plants that have not been genetically modified. And if you look on either side of that, you'll see a not so random string of numbers and letters that denotes a transgenic variation of the rice plant. So what this image is showing us is the results of an experiment where transgenic rice plants and non-transgenic rice plants were subjected to drought conditions, where essentially they weren't watered for a number of days, and then they were re-watered for a few days, and they observed the following results. Essentially, the rice plants that had been modified showed significantly less stress symptoms, and they recovered more quickly once they began to be re-watered. If you look on the right side of this diagram, these bar graphs, what they're showing are measurements of chlorophyll fluorescence in transgenic and non-transgenic plants. You can see that all the colored bars are transgenic variations. You can tell they're the transgenic variations because they are the not so random string of numbers and letters. And in you can also see that the white bars are NT or non-transgenic. And basically what this shows us is that the transgenic varieties of rice showed significantly higher chlorophyll fluorescence than the non-transgenic variations on average, given a number of abiotic stress circumstances. Why is chlorophyll fluorescence important, you might ask? Well, chlorophyll fluorescence is a really good indicator of the general well-being of the plant and how much it's photosynthesizing. And so in total, these results tell the scientists that their modifications are working. And an important aspect of these results that isn't shown by this diagram is that things like the breeding ability of the plant are conserved, and that many of these changes are passed down to the next generation of plants. And so this is really important for industrial applications because obviously they need to be able to breed the plants and, excuse me, they need to be able to breed the plants and, sorry, I'm blanking a bunch. Basically, the breeding capabilities of the plants are really important, and these are conserved in spite of the genetic changes that have been made. And so this is just one example of the sort of results that we might see. But there are a number of other studies that give us similar results that show that plants can be targeted for very specific modifications without negative downstream effects. And so this is why gene editing can help us confront climate change so effectively. Essentially, methods like CRISPR-Cas9 dramatically reduce the amount of time and equipment required to make genetic modifications. In more conventional circumstances, you would have to breed many dozens, if not hundreds of generations of plants together in order to bring about desirable traits. And this often involves back crossing, which involves inbreeding. And so if we were to try to select for desirable traits phenotypically, we wouldn't necessarily know what's happening on the genetic level of the plant in the same way that we would if we were making changes mediated by CRISPR-Cas9. Additionally, the changes that we make using CRISPR-Cas9 don't depend on us having a pre-existing variation of the plant with a desirable trait to breed with another variation of the plant. And so essentially, this just dramatically reduces the amount of equipment and the scale of genetic modification projects. And so where can we take this technology? This technology, CRISPR-Cas9 and agrobacterium-mediated gene transformation, or agroinfiltration, as Umair referred to it, these genuinely have the capacity to change the world. And unfortunately, the barriers that exist to their application are preventing these technologies from being used to their highest potential. At the moment, we're studying things like, can we modify the plants? And are the changes that we make safe and effective? And the results are, I mean, so far are pretty great. That being said, there are more profound applications of these technologies. Like CRISPR-Cas9 may one day be used to cure all manner of diseases. Or in the case of addressing climate change, we could potentially modify plants to sequester more carbon, not just mitigating climate change or increasing yield under climate change circumstances, but potentially reversing it entirely. But this depends on us having a much more effective and internationally consistent regulatory framework that's more in touch with where the scientific consensus is on genetically modified organisms. And so until that happens, we can't even really begin to work with these more profound applications. That being said, hopefully with a little bit of time and with a little bit of science communication like this, we can build a deeper trust in the technology and maybe eventually get around to doing some really, really incredible things with it. So thanks for listening to my presentation. Oh, whoops.
That was great. Thank you.
Oh. Would somebody mind getting me out, because I am currently lost in the menus.
Jeanette, are you able to abort the screen share?
Whoops, sorry.
All right.
There we are.
I think I've done it.
Okay, sweet. Jeanette, did you want to lead on some questions? And
Definitely.
I have some too, if you run out or whatever, I'm happy to.
Yes, absolutely. And I wanted to start with a question that isn't directly related to the content of the presentations tonight, but I thought because we have students here and they are captive and I can ask them my questions, there might be people in the audience who don't really know how Quest works, how it might be different from other schools. And we have many times had that explained to us by instructors, but I would be really interested to hear from the students' perspective what it's like to be a student at Quest. Would anybody like to jump in? And of course, you can talk about pre and post COVID experiences, because I think all three of you have experienced a bit of both. Anybody?
I guess I can start off with this if no one else minds. I think kind of to try and answer your question, I was actually a transfer student into Quest, and I can definitely compare it to past colleges. So I transferred to Quest out of college in Florida, studying actually aerospace. I'm hugely interested in engineering and aircraft and all kinds of sciences, but I was really frustrated because I was so locked into one path. I had friends from all different disciplines, and they were all doing really interesting projects. I was like, oh man, that seems really cool. I'd like to be involved in that. But my class load is so strict and set forward that I didn't have the option to do that. Transferring to Quest, it isn't based off of some major where you have all of your classes set out for you. You have a question, and your question can be anything that you're interested in.
So my question is, how do we engineer urban ecologies is pretty carefully chosen to be about engineering, but I get to combine all of the projects and things I'm interested in. So I can look at civil engineering with, different concrete and ways we, lay out our roads and buildings. But I also get to look at, biology and ecology and other things like that that really interest me that at my old college I never would have looked at. I think this is certainly carried over even into the COVID experience with online learning. I think that even though, it's definitely, as a student, it can be difficult because it can be mentally taxing, often to sit at a screen all day, every day, usually in like one or two places, maybe around your house. I still get the option, and we can include so much more. In this class, we had an entire chat that's just like cool things about plants, and I can relate this to other things that I find online. So you can continue that interdisciplinary looking at things, and it's like, oh, if I wanted to talk about, the ways in which we can modify plants and fruits so that they can survive in hot environments, that's something you can do. And I feel that being online in no way hampers your ability to be, in a part of a whole bunch of different disciplines.
Awesome. Yeah, I mean, that sounds great. It sounds like there's quite a bit of freedom to follow your own question, which is pretty amazing. Now, and any of you can jump in here, or Marina could jump in as well. What is the process like for choosing your question? Do you have a supervisor that helps you? Is it Marina? I don't know. Does somebody help you in your first? And I know you don't have traditional semesters either. So does somebody help you choose your question? What's that process like? Anybody, go for it, Kenya.
Well, it's pretty much entirely self-directed. That being said, there are some steps that we go through in order to solidify our question through, I guess, more through a framework meant to produce a certain degree of specificity. We want to bring about a certain degree of quality in the kind of question that somebody's going to pursue. We want to make sure that it isn't just, I mean, ideally it's the question that they want to ask, but that shouldn't be, for lack of a better phrase, an empty question. You have to define what the boundaries of that question are, and you have to set forth plans for how you're going to go about investigating that question. And so while you have quite a bit of time and space in your first few years to figure out what it is that interests you, in the last 2 years of your schooling, you really have to begin to specify, and there's specific classes like Question and like Keystone that are meant to have you arrive at specific outcomes, not necessarily in terms of what it is that you're asking, but what you might produce in asking that question. And so it's really open-ended, but there's obviously certain requirements that need to be met. And so it has to be balanced in some ways. That being said, the open-endedness of it is, in my opinion, the most appealing quality of the whole question affair. So, oh, and I forgot to include, there are a number of people who will guide you along this process, including instructors and fellow students. So at no point are you alone and just left entirely to your own devices in trying to process what might constitute a good question.
Thank goodness for that.
Yeah, and if I can add one thing, is that you also have to do a sort of exam. It's kind of like a mini version of a qualifying exam that you would do for a PhD project, but you have to sort of have a meeting with your mentor. So you choose somebody who is a faculty who will, in theory, support you, hopefully, in pursuing your question. And they chair an oral exam where you essentially defend your plan. And so that's meant to happen shortly after taking that question course.
Well, and that sounds like a great experience, because presumably that could prepare a student for entering a conventional grad school experience where you will have to do candidacy and things like that. Awesome. These are things I've never known about Quest, so I'm very, very interested to learn. Moving on from that, okay, so one thing that I feel like all, not just scientists, but academics have to develop is their elevator pitch. You know, if you were, now, not that we get in elevators with people anymore, come on now. But, the idea of if you were riding an elevator with a stranger, how would you sum up your project to them, assuming they know nothing about your field, in, you know, the 2 or 3 minutes that that it would take for you to, you know, get up to the top floor of that skyscraper? I'd be so interested to hear if any of you have an elevator pitch for your question. Does anybody want to take a stab at that?
Well, I'm afraid at this point that elevator pitch hasn't quite materialized, as my question as well has not quite materialized entirely. I'm still partly through this process, and so I'm developing the elevator pitch as we speak. But as it stands, I'm extremely interested in essentially how physiologically we should go about nourishing our bodies, and I want to pursue that through some sort of study of how we distribute our nutritional resources. And so while I don't have an elevator pitch for you, you know, these things are coming together slowly but surely. And I'm still in the middle of the process, so.
That's a good hint at what your speech may be. Does anybody else want to take a stab at that?
Yeah, so I think I might be a little bit further in the process than Kenya. I've actually done a lot of this. One of the things you're required to do for some of this examination is, like, I end up giving a little talk about my question a whole bunch to people. So my kind of elevator pitch is, we do an enormous amount of research in our fields and forests and natural, you know, surroundings. But unfortunately, our urban spaces are expanding at a massive rate. Cities can be, you know, tens or even in some cases, you know, entire islands of just city and urban space. And we don't understand fully all of the organic interactions that are taking place here. A recent study showed that house cats have enormous impacts on the ecology around them. They're massively ecosystem altering, and we plant trees in our roads and stuff that we don't understand if they belong there or anything. And so in order to begin to understand, you know, the nature around us, I think it's important that we look at our cities and in the spaces that we're creating and controlling, and we understand in these human created spaces what all the interactions between all these organisms are, how we can study them, and how we can start to make our cities places which are not only cities, which are livable for humans, but also healthy ecosystems in their own right.
That's not a bad pitch right there. I think, Kristen, you touched on something that I think is important in communicating in the sciences to the public, which is, why should I care? You know, it feels very cold saying that, but I feel like, you know, Joe Public is probably going to ask you that about your work. Why should I care? How's it going to impact me? So I think you've done a great job of I don't know, providing context for why someone would care about this work. Umayyar, would you like to jump in? Have you started working on a pitch?
Not really. I kind of am doing this wrong. Like, you're supposed to only do foundation classes in your first 2 years, but this is like my 5th concentration in a row, so I don't have a question and I don't have a pitch. I have a working question, but I don't have that approved. I'm hopefully taking the question block sometimes in spring, and hopefully we'll figure that out then.
I wonder though if you could comment on you mentioned in your little spiel to me that you're possibly interested in genetic counseling, and that's probably something that people might not know much about. So I'm wondering if you could just really briefly say why you would be interested in that and or what that is.
Right. So genetic counseling is
kind of new. It's not really offered in a lot of places. I know that UBC has a program here in BC, and I know that Toronto has a few programs. But what genetic counselors basically do is study your genetic profile and work with the doctors to suggest the best possible treatment for you. And they're also kind of a counselor, like your emotional counselor, because genes can be very tricky to talk about, especially in cases of like you might have a gene that you don't, that might be bad news, and you need someone to talk you through that and to basically break that news to you gently and explain to you in a way that will hopefully make your life easier. And I think that kind of gives the whole interdisciplinary education kind of a whole different meaning, and I can like explore all these different things, and hope that is just one of the options, and that's something I'm interested in, but I'm not sure if I'm going to do that. But yeah, and like I kind of have Marina to thank for that, because I took this class with her, and she brought on this like class of genetic counselors who told us about what they do and how they do it. And yeah, that's kind of what got me interested in genetic counseling to begin with.
I mean, that's such an interesting potential career path. And I imagine, well, I know when Marina did a talk about epigenetics several years ago at the library, and I was so fascinated by that topic. And I think genetics is a branch of biology that really the public doesn't understand much about. So I feel like the role of the counselor would be so crucial, because like you said, it could be bad news for you, but I guess it could also be bad news for your offspring, your family. So certainly it could be a very complex procedure to go through with somebody, to have to unpack all of their genetic baggage, so to speak. I've actually had a question come in, and from one of our viewers, and it says, this is a great one. Were you plant people before you started at Quest, or did your sort of specialties bring you to plants? I'm very curious to hear about this.
I'd like to preface this by saying I have never, until fairly recently, been what I would describe as a plant person. As you can see behind me, I have recently adopted 3 little plants, and I'm a newly minted plant dad as of this block. And so while initially I wasn't particularly interested in plants, I think in general, in my general studies in my first few years, I brushed up on my ecology a bit, and that put me in closer proximity to plants, forced to think about them in a more meaningful capacity. And I even then, because animals, particularly large animals, tend to be a little bit more charismatic than plants are. They get a disproportionate amount of the attention. So even though I was studying ecology that heavily involved plants, I still didn't quite give them their due until this course, and I was really forced to recontextualize my understanding of plants quite a bit. And frankly, I can't stop looking at plants. It sounds a little bit corny. I'm not being paid to say this. Every time I go outside now, it's really hard not to think about all the plants that I'm passing by, just because I've been forced to realize just how much is happening in them and between them. And while my interest is a little bit more in human physiology, as somebody who's enthusiastic about biology broadly, I say I really like plants now. I mean, I might even say I adore them. They're absolutely fascinating.
A beautiful tribute to plants. Anybody else want to weigh in on their plant love? Yeah, go for it.
So growing up, my grandma had a garden in our roof, and she still does, but I haven't been home in a very long time. And even like growing up, I did not realize how much effort it takes to actually grow a garden that's going to be healthy and it's going to exist as long as it has. And I never really appreciated plants because it was always around me. And even like coming to Squamish and like living at Quest, there were forests around, and I always appreciated how beautiful it was, but I did not really like stop to look at a plant or anything. But then I took this class at Quest called Science of Health and Wellness, where I did this research project with my group that involved plant medicine. And that's what initially got me interested in plants. And then I took What is Life with Marina, and we did this paper called where they described how plants communicated with each other, which I thought was very fascinating. So like when I saw that this class was happening, I decided to sign up, and it made me realize like how much there is to plants and how much we do not really realize and how much we do not see, like Kenya had said. And yeah, now when I look at a plant, I'm like, wow, like there is so much going on, and you can't really tell that, but it's great because especially because like you think that, oh, it's just a plant and it can't move and it can't feel and it can't see anything, and but it does all those things. And I was actually super surprised to learn that there are no ethical laws about genetically modifying plants as there are about genetically modifying animals, and I guess that says something about our society. But yeah, I really appreciate plants a lot now, and I'm glad that Quest kind of lets me explore all these different things that I never thought I would.
That's awesome. Tristan, are you going to continue the plant love?
Sure. I mean, I think first of all, it's actually really hard to be in British Columbia, to be in Squamish without falling in love with the forests. It's, you know, it's pretty much like there. You can't go to Quest and like ignore the trees. But I think I also certainly wasn't a huge plant person before this class. I spent most of my time in shops doing a lot of, you know, machinery stuff and engineering stuff. But I think it's one of those things where something we talked about in this class, and we've talked about, I've now taken quite a few classes on plants. We talk about plant blindness, where people, you know, live their entire lives surrounded by plants and manage not to really take notice of any of them. And it's one of those things where like, once you get a little bit of curiosity and you start to like look into it, it's just like a huge like ocean, like you can dive and like never see the bottom of like the plant science. So there's just like, I guess it's like a force, you can just get lost in it. It's really great. So yeah.
It would have been a really bold move if you were like, no, I hate plants. But I'm glad you love them as well. You're so right. I mean, it's kind of impossible to be in this part of the world and not be just in awe of what is around you. I moved from Newfoundland to BC 10 years ago, and just the scale of the trees completely, you know, knocked me out. Now, switching gears from plants, I have another question from the audience. And it's kind of going back to the Quest experience for the students. And the question is, do Quest courses involve field or lab work? And if so, what are you doing about that during COVID? This could be a Marina question as well.
I can certainly start off and just say that yes, both, and yes, weirdly. So I think our faculty, for the most part, pride themselves on trying to incorporate some kind of experiential learning, whether that be field-based learning or lab-based learning. Yeah, like we have some pretty epic field trips that happen that are like multi-day field trips to visit volcanoes, to go to Vancouver Island and learn about, say, like island biogeography and marine habitats, to like day trips to the aquarium or to a local forest or a local estuary. So the field trips really sort of run the gamut. I know my friend and colleague Tor takes students down to Peru, for example, to learn about tropical ecology. Anyway, and then in the lab, we try, we don't have a state-of-the-art research lab since we are not a research institution, but we do have a lab and we are trying to grow capacity as best we can within there. And so we typically do offer lab-based learning for things like molecular biology, biochemistry, and chemistry. And then we also have an exercise physiology lab where my colleagues, Molly Welsh and Megan McNutt, do exercise physiology type testing and things like that. So yes. And then in terms of COVID, it really runs the gamut. Some people have opted to transition to largely bioinformatics or like data-driven existing data sets and analysis in that way. Others have done like choose your own adventure style field trips or guided field trips. Like they go on, like the professor goes on the field trip and has video. I know my colleague Emma has recorded herself doing chemistry labs. I have recorded myself doing labs, quickly realized that I am not a movie star and instead decided to, in this class, for example, just ask students to tell me what to do. And then I do that and I collect the data and post it for them to analyze. So it's definitely a bit clunky, and it's not, there's certainly a loss in terms of hands-on learning and technical skill development. But I think most of us have tried to opt towards trying to build analytical and science communication skill development in lieu of the hands-on. And I know I certainly, and I think Emma as well, are planning to try and have some remedial activities once we are back in person to offer students an opportunity to get some of that hands-on learning that they didn't get while we were online.
Awesome. I mean, you make a good point that, analytics and communication are crucial. And so, it doesn't sound like a total bad thing to be spending more time on those subjects because, and certainly every academic in every field needs to understand those two aspects of their work. So even if they might not want to, do that stats course or what have you, it will behoove them to have those skills.
No, nobody wants to do the stats course. Let's be real. Nobody.
Alas. Marina, you said you have some questions.
Yeah,
Would you like to ask a couple?
Sure, I could ask a couple of questions that are a bit more geared towards the presentations. They're kind of similar, I think. I have a question. So, Umair, I'm wondering, I remember you mentioned early in your research process and that you were having difficulty finding papers about vaccines and other sort of like farming with a PH in plants. And I'm wondering if you might comment on, given that you now have tried to review the literature as best you can, you've written a research paper, you've made a presentation. I wonder if you have any ideas about why that might be. Like why isn't there more research? Do you have any feel for why there might not be more research on this and if it's coming?
So one of the biggest issues that I had when researching vaccines is that plant vaccines do not get approved by the FDA, and it's really hard for people to get plant-made pharmaceuticals approved. And that's why, because like the assignment prompt was to do something that does not already exist, I chose to like look at it from the point of view of the process itself, of the process of transforming the plant to make those proteins. And I kind of found that a bit difficult as well, because I found that a lot of the research were being done by the same team again and again, and they were trying to like convince people that this is something that can be cool and this is something that works. But it seemed like not a lot of people were picking up on their research or doing research of their own. So it was like the same authors again and again. And I think it's basically has to do with plant blindness and people not trusting genetically modified organisms making pharmaceuticals, even though they take insulin made by bacteria for some weird reason. I don't know. But yeah, there's definitely the plant blindness fact involved. And also there's not a lot of research being done in plants, and not a lot of plants have been optimized to produce pharmaceuticals in them because there is not a lot of research going on. And there are a few companies that I know that are working on making plant-made pharmaceuticals, but none of them have been approved by the FDA as of yet. I only know of one plant-made pharmaceutical that has been approved by the FDA, and that's this therapeutic enzyme. And everything else is either in preclinical stages or they're going clinical trials, but they haven't been approved yet. But because of the recent pandemic that we're in right now, the COVID-19 vaccine is being quickly tested for, and hopefully that will lead us somewhere.
Cool. Thank you. Yeah, great. I had another question that's on a similar line of like, how come, for Tristan. Tristan, so you talked about how there hasn't been that much research in terms of genetically modifying plants to deal with heat and climate change. And I also remember we had a guest lecturer a couple weeks ago now from Syngenta, which is a really big time GMO sort of player. And they too also pointed out that there hasn't been a lot of this. And I'm wondering, again, from your readings and from your own impression, if you have any idea on why this might be the case, because it strikes me as really strange, given that A, we've known climate change is happening for a while, and B, we know that there are crops that are potentially going to disappear if it gets too hot. For example, and not to sound too anxious or worried, coffee, right? Like, can we imagine a future without coffee? But coffee has like a really specific environment in which it grows, and that is thought to be disappearing quickly if climate change continues unchecked. And so it just strikes me as strange. And I'm wondering, you may not know, but it was more of a like, this is weird. And do you have any ideas on why this might be the case? Like, why has nobody been working on this?
Yeah, so I'm definitely on the boat with you with coffee. I'm also freaking out. But I think that there's a couple of different things going on here, and each of them could individually kind of help to explain. I don't specifically know with coffee, which now that you've brought it up after this talk, that's number one priority. But I think, so the first major thing is there is research being done onto how to make crops and fruits and everything kind of be able to grow in a warmer climate. But a lot of it isn't genetic. One of the things that I found over and over again was research on like, here's some new fertilizer you can use, or here's some shading technique, or here's some other agricultural technique. And that popped up over and over again. I think people are treating genetic modification in a lot of cases as a last resort for keeping their crops alive in warmer climates. And I think that that feeds a little bit into my second point, which is a lot of people just don't want to eat GMO foods. I think there's been a lot of push for people to like at least get, you know, everything GMO like labeled, and very much so like, no, I don't want any GMO in anything that I eat. I don't want to get too in detail on that, but the research pretty much says, well, there's no big deal, but if that's your feeling, then, you know, you eat the food that you want to eat. But I think that because there's, you know, a lot less incentive for people to eat GMO food, companies don't want to put time and effort, you know, producing a crop that then everyone's going to turn their noses up at. Lastly, again, with the people producing crops, I think there probably is research in this. I'm pretty sure that people are doing research on apples to make sure they're heat resistant. But a lot of this is some company being very quiet about how they're doing this because they don't want some conniving college kid to write up a report on how you can make apples resistant to heat and then steal a lot of their profit. So I think a lot of stuff that's going on also, you know, genetic engineers are keeping under wraps so that competitors don't try and swoop in and take all of that. I think those three factors kind of combined kind of lead to things, which I personally think is a shame, because if we lose coffee, I don't know what I'm gonna do. So I think that we should probably get on it, and we're really close, but just a couple of things are barring us from getting heat resistant fruit and crop.
You raise an interesting point in that there's the proprietary issue as well. And this is going to perhaps lead into a question for Kenya as well. But I'm struck by the fact that an outcome of plant blindness is that we know there. I should have pulled the stats and I don't have them, but there are stats to demonstrate that we are graduating fewer and fewer biology majors specializing in, say, botany or molecular plant biology or whatever. And this is quite striking. I had a mentee a couple years ago that was entirely focused on this for her question, but it's quite striking because we basically are decreasing the number of experts, and yet we have an increasing demand if we think about the challenges that these students are just outlining for us in terms of climate change, food security, the need to grow a lot more food, and yet we have fewer and fewer people graduating to populate all these jobs. On top of that, plant blindness is also impacting grants in terms of whose research gets funded. And so this is me totally armchair philosophizing here, so please don't quote me as like an authority, but I wonder how much of that plant blindness factoring into grant decisions has actually really depressed research in the academic nonprofit sector on these kinds of solutions and pushed it into the proprietary sector. And there we don't necessarily hear about it, which then might exacerbate those feelings of distrust in terms of who is actually generating GMOs. Some of the first GMOs, and I think this is actually still true, that were developed in research labs in universities by professors that did not have profit stakes in these. And yet, nevertheless, there's this impression that it's all about like big conglomerates like Syngenta and Monsanto. So that leads me to a question for Kenya. When we think about things like CRISPR, one of the things that's really exciting about CRISPR, and maybe scary for some, is that it's so easy and so cheap that I could just order my own CRISPR kit and make my own genetically modified plants using CRISPR with like relative ease. Maybe not as much as like in a lab, but still relatively easy. And with the onset of CRISPR, there was a huge uptick in DIY scientists, which are becoming quite popular. There was like these hilarious videos of people like CRISPRing themselves on YouTube and things like that. And so CRISPR has this interesting feature. By the way, CRISPR just got the Nobel Prize, FYI, in chemistry, that in some ways it's democratizing the accessibility to things like genetic modification. And then it's also in some ways stealthy, right? And so we actually already have CRISPR edited products on the market that are not regulated, right? So like they will not be labeled as GMOs. And I wonder, Kenya, what you think in terms of, does that potentially cause a concern in terms of like, if this is stealthy or whatever, maybe I'm just creating a problem here by calling it that, might that actually exacerbate distrust? If you know what I mean? I don't know if I'm being coherent here.
Yeah, I think I can speak to that. I think the shame about the situation with such immense public distrust is that I think the distrust that comes from lacking an understanding about how the technology works, and a distrust that comes from, frankly, a healthy distrust of large corporations and their usually selfish interests. That distrust has been pushed onto scientists who go through a really painstaking effort to reaffirm the accuracy of the results. And in the case of a technology like this, how safe it is to use, what it's like, so much time and energy is put into figuring out what its downstream effects are. And I have a great deal more trust in scientists than I do, say a corporate interest. And so I would frankly prefer that scientists and people who I know have made it their life's work and are qualified to be administering these sort of technologies are the ones working with them, ideally with some sort of public oversight. I would prefer that, frankly, to people who don't have a great understanding of the technology, somewhat implementing it blindly or brazenly. And so while the democratization of this kind of technology, I think, is incredibly important, and everybody should have control over themselves, I am also equally concerned with the general well-being of people. And so I think this is something where there needs to be a unifying regulatory framework. Again, like I mentioned in my presentation, because there's this lack of understanding about how the technology itself works. I don't think we're going to arrive at the point where we could comfortably allow just anybody access to this technology, because we just don't have the civil protections in place, and we don't fully understand the technology, but the scientists are putting in all the work to figure that out. I have faith in them and the work they're doing, and so I would rather it be left in their hands than we release this too early, or we allow corporations to use it as they see fit. I don't know if that answers precisely what you're trying to get at in the question.
It gets at like the trust issue, and I guess I'll just like add again, like I guess one of the things that I think is interesting and paradoxical is that because CRISPR-Cas9 is such a surgical tool, because that means that then these products of CRISPR-Cas9 editing are not being regulated as GMOs, that like we already have fish, for example. There's fish on the market in your grocery store right now that are CRISPR'd fish. I have no problem with that, but I could see how somebody who is anti-GMO learning that I wonder if it would exacerbate. And it's interesting. And I guess one thing, and I know we're running out of time, but one thing I think that it's worth sharing for like whatever it's worth for this pedestal soapbox that I have at my disposal right now, is that probably the thing that will stuck the most with me from this course was the realization from our guest speaker from that biotech, that he pointed out that essentially in Europe, where the oh hi Raven, where the regulations are quite strict with respect to GMOs, that they basically use genetic modification tools for research to get to a product that is what they want and get there quickly, and then they use old technology to get there again so that they can actually disseminate that product commercially. So to me, that is so backwards. Like I feel like there's an analogy there to use, and I can't quite think of it, but it's I don't know. It's mind-boggling to me that that's where we're at, where you have such a distrust of a technology that people are using it and then using the old technology to arrive at the same outcome. Same outcome is the key thing there, and then that's okay. It's mind-blowing. And I feel like that is something that is important for people to appreciate, that is what's going on, because that is silly. That's Marina's little rant for you.
CRISPR'd fish may sound delicious.
I mean, crispy fish certainly does. I can get on board with that.
Well, okay. I hope you're all still, listening in. That was Marina's impassioned offering to our audience. And of course, it wouldn't be a Zoom call if a cat didn't come in to the screen, and maybe a partner also. That means that this is a real authentic Zoom call. And it is 8:30, so our time is up. I want to say thank you to all of you again. This was so enlightening, and I have so much to think about. I had another question about going back to the charismatic species and whether there were like sexy plants that people wanted to study. I guess that's a question for another time. Once again, I will thank our friends at Squamish Public Library, at the Pemberton Public Library, and on behalf of Worcester Public Library, I will say thank you to Quest and all of our speakers. And we are going to say goodbye to our Facebook friends right now. So goodbye, Facebook.